मुझे लग रहा है डॉक्यूमेंट भी रखने पड़ेंगे, जो पास, ट्रांसलेशन जितने भी हैं वो सारे डॉक्यूमेंट रख लेना।
डॉक्टर ज्योति, आप रेडी हैं?
Yes, sir.
ठीक है, प्लीज आप प्रोसीड करिए, पार्टिसिपेंट्स पर ऐड होते रहेंगे।
A very good morning to you all. Today, I, Dr. Jyoti Sharma, welcome you all in 9th day of International Winter School come training on. Am I audible?
हाँ, यस प्लीज।
Okay. A very good morning to you all. Today, I, Dr. Jyoti Sharma, welcome you all in 9th day of International Winter School come training on navigating agriculture transformation for upliftment of rural excellence. That is Nature 2020-24. Today we have with us eminent speaker Dr. Vishnu D. Rajput, working as principal scientist and head at International Laboratory on Nanobiotechnology at Southern Federal University, Russia. His ongoing research is based on the investigation of effective remediation approaches using biochar, nanobiochar-based solvent and nanomaterials. With long experience and experimental work, Dr. Rajput comprehensively detailed the state of research in environmental science about how nanoparticle or heavy metal interact with plants, soil, microbial community, and the larger environment as well. And also he gave the remediation technology using nanoparticles oblique nanobiochar. In recent study, he is working on nanobioengineering for soil rhizobic microbes and enhanced remediation using nanobiotechnology, CRISPR and metagenomics. Dr. Rajput is included in world top 2% scientists and obtained second top rank in environmental and earth science in Russian Federation. He has published more than 300 research papers and have 53 H-index score. During the last 5 years, he has received many appreciation letters by Rector of Southern Federal University for breakthrough research in environmental and soil science. Sir has done a lot of research in the field of nanotechnology. So, sir, on the part of AgriMeet Foundation and World Agriculture Organization, I welcome you for the today's lecture. Sir, please.
So thank you very much. Greetings from Russia. Thank you, Dr. Ankur, for inviting me. Thank you, madam, for introduction. And it will be really great pleasure to talk about our research. My throat is not good today. I have cough, but I hope you will manage to listen me. So before going, once again, thank you to all of you for joining. And I'm very sorry for changing the schedule time because due to some another engagement. And thanks, Ankur, again. So the talk devoted today about nanomaterial, nanobiochar, soil health, and crop production. Hope my slide is okay.
Yes, sir. Yes, sir. Please proceed.
So, you know why this research is significant in current scenario. We know or last several years, I can say like 4 to 5 decades after Green Revolution in India, even here in Russia, in China. Why I'm talking about these 3 countries? Because I am Indian and I study there. My whole education in agricultural background, and I worked also 5 years nearly in China. So I know their soils and their agriculture. And here in Russia, I'm working last 9 years almost this year. So it's long experience for all these 3 countries. So you know these 3 countries have like both India and China is populous country, and even population in India now increasing rapidly, and also agricultural countries. In Russia, it's not only cold, but if you say south part of Russia, we have very good soil. It's called Chernozem, Haplik Chernozem. Chernozem means it's a black soil. It's a Russian word, Chernozem. This soil have nearly 4%, sometimes even more organic matter content. But if you see the ratio or average of Indian soil, even some soils having less than 0.1% organic matter. So why organic matter is important? Because if soil is rich in organic matter, definitely it's a good for production, crop production. So, you know, the challenges now facing to improve soil health, to achieve zero hunger goal that is also set by United Nations, especially I can say these populous countries, in a sustainable way. So what is the technologies right now? So that we are going to discuss. Also soil facing several other problems like salinity problems, drought stress. We also have drought stress, drought problem in south part of Russia, but salinity is not big issue in Russian Federation, but in India, because I also worked in Central Soil Salinity Research Institute, so I come to know that many places they have reclaimed the saline soil, but it's still because secondary salinization and other processes is going on, so the soil getting more and more saline. Problem here we can say this is human or anthropogenic problem, like metal stress, elemental stress, toxic elements, emerging contaminants. These are the terms which you might be hearing every time. Now nanomaterial also considering as a emerging contaminants, microplastic, nanoplastics, So also heavy metals because of rapid industrial technogenic activities, anthropogenic activities. So toxic elements directly, indirectly coming to the cultivated soil is going to the environment, especially soil becoming a sink for this all toxic elements, and directly, indirectly, of course, it affecting human health. In the last part of this slide, here you can see our rhizospheric microbiome zone is significant, very important for crop production. Sometimes we know that some soil rich in particular element, for example, phosphorus, but it is in unavailable form, not rich in microorganism. So even the nutrients are available there, but plant is unable to access those new elements. So this stress, I called it soil stress. This also affecting our crop yield. So last even 7 decades or 6 decades, we are continuously applying chemical pesticides, chemical fertilizers. So the excess amount after crop production is remain in soil and damaging soil biota like earthworm, many organisms, agriculturally important microorganism. But it also damaging this rhizospheric microbiome, which is responsible for plant growth. So continuously use, and now soil is saying that I cannot produce anything without additional elemental support. So it means the soil is now cannot naturally restore or cannot naturally produce anything until unless you have to apply something, some fertilizers. So this is a alarming issue because you cannot continue this practice further many years. Here some approaches, I explained some approaches here like nanomaterial, biochar, nanobiochar, and microorganism. If you see in second part, this part I showed is like a hyperaccumulator plant, like phytoremediation, bioremediation. These terms are also not new. Even we are working last several years about phytoremediation, bioremediation. I did these approaches like phytoremediation. What is this? That when you grow plant, those plants are a specific plant, have capability to accumulate toxic element from the soil to their body, their tissues. So it's like phytoremediation. Also the microorganism, those who can stabilize or convert the toxic element in non-toxic form. But these approaches, the phytoremediation and bioremediation, the long researches, still we do not have any realistic technology which we can access easily and environmental friendly and sustainable low cost. So this we are still suffering. So year 2000 it's like a nano era started. So a lot of laboratories started working on nanomaterial. They are in various ways. But why it is interesting? Because at nano scale, the material have different properties, different behavior, different chemical reactivity. So this is very interesting for several fields like agriculture, for crop production, for medical, pharmaceutical, also making even in automobiles. So you make nanomaterial from zinc, it might be transparent. So a lot of beneficiary applications for nanomaterial. So last 20 years, a lot of researches on nanomaterial. I also started since 2016 working on nanomaterial, but my task not related with the synthesis I try to understand the mechanism of nanomaterial, how it going or translocating from root to shoot, foliar application, leaf to root, how it translocate within the cell, how it mobile, where it accumulate, what concentration it accumulate, what form is accumulate, like mechanistic part. So the target was to synthesize such type of material which we can use as a targeted material. Like if we want to express this material only in particular place, like in photosynthetic part, then this material express in that place. So also we worked on biochar. So biochar we working on last 5 years. So biochar is also not a new term, but why it is interesting in current scenario? Because a lot of other amendment, even you see this term biochar is a carbon, but you see this term for many years, even in literature you can find. But in this era, I mean the current scenario, it become really popular. Now, like a nanomaterial, many scientists working on biochar. Because of now we have several other soil amendment or plant growth promoters, plant growth enhancers. So we are combining those soil amendment material with the biochar. Like we are making modified biochar. We are also know that at maybe nano scale, like nanomaterial at nano scale have different reactivity and chemical properties. Maybe biochar have similar beneficiary impact. So that's why now we are combining biochar with metal organic framework. This is the material with nanomaterial, with specific bacteria. If you see in last specific bacteria, the bacteria which can we use as a bioremediation. It means like some specific microorganism that are helping to reclaim particular soil polluted with the specific elements, or those microbes sometimes helping with multi-metal polluted soil also. Not only metal, I mean organic and inorganic, both pollutants. So here that's why we try to combine this approach. We taken a traditional methods like hyperaccumulator plants and microorganism. These microorganism we also developed in our laboratory. We have, I mean, we isolated and screened, and we use with biochar. And now biochar can be produced from anything, I mean from different sources. We are last project I have completed with BRICS. It was with India and China, 3 countries. In this project, we targeted food waste biochar. So we collected food waste from 3 cities of Russia and prepared biochar, because food waste is also a problem. So if we can use or we can convert as a value-added product and we can use in agriculture, it's very interesting and solving many. So this approach I showed here, like how we can go for the nano remediation or phytoremediation, where we can use these materials and enhance your traditional approaches. Biochar also interesting. Why? Because biochar is a porous material, and combined with microorganism, it provide very good habitat to microbes. So if you reduce the biochar at a nano scale, it have or it enhance the more beneficiary impact, especially for microorganism functionality and for soil health improvement, because the surface area increased. Similarly with nanomaterial also, because a tiny size surface area is increased. So with microbes it's have good impact. But of course several kind of nanomaterial also have negative impact on microorganism, because we are using as a pesticide or fungicide also. So, you need to find the specific microorganism and also a specific biochar. because some biochar, for example, you produce biochar from sewage sludge that contain high amount of metals, elements. Heavy metals, I mean, this is also essential for plant, but when it exceed the limit, then we call this a toxic elements. Yeah. So when you prepare biochar, it is necessary to characterize your biochar and understand the state of biochar, which type of elements available, and then you apply soil. You cannot apply random any kind of biochar in any soil, because some soil are rich in and deficit in some particular nutrients, so you can apply such type of modified biochar in that soil. I already said that several kind of biochar like metal biochar, mineral rich biochar, yeah, double layered rich biochar, these are the materials which are already using in soil remediation, microorganism. And here you see here we develop a technology in our lab, like fabrication of biochar derived from different type of feedstock. Feedstock is like a source material. So we try to standardize, because of course it can be a variable with the source material. So but we taken like rice straw, wheat straw, and also from the wastage, agriculture wastage, and then we define what time, holding time, and what is the temperature will be better to pyrolyze biochar, because we try to convert material without under anoxic condition, and then biochar generated. And biochar also stabilize carbon, so it helping in carbon sequestration. We are not directly working on this aspect, but I have a small work we just started about carbon sequestration also. But the mechanism is more interesting, where you are using all these three variant, microorganism and nanomaterial. Then this materials we are using like a amendment, like a enhancer the process. So I already told us, since 2016 I started working on nanomaterial. Main target for this experiment was to understand the mechanism, how nanomaterial behave on soil microorganism or on plants, inside the plant body, and on plant physiology. So we tried like several kind of experiment, even I can say series of experiment. Some experiment we did in hydroponic condition, hydroponic like growing plant in water condition, giving specific nutrients and conditions. So this is interesting, because here you can see realistic impact, what you exactly want, because all conditions are controlled. In field condition, the situation is different because environmental factors. So we also try in model experiment, where we have different even we treated nanomaterial as a pollutant. We use nanomaterial as a foliar spray under the multi-metal polluted soil, to try to understand either application of nanomaterial as a foliar or as a vessel can alleviate the toxicity of heavy metals. We have group, so other people also work venjaprine, yes, pesticide, but I'm not directly working on that aspect. So before understanding this, it was necessary, because this is a previous work, and we are continuously working on the further aspect. So we first try to monitor either nanomaterial really accumulate in plant cells, plant tissues, organs. Yes, so we not only, because how we analyze in traditional way in lab any kind of elements. So mostly what we doing, we are taking any part like leaf or stem, if we want to observe how much concentration of some elements, and then we grind and go for the digestion method. So here we also use approach like physics techniques. So we use like transmission microscopy, electron microscopy. Also we go for the X-ray fluorescence, and we go for the XNAS. It's kind of synchrotron methodology. And the interesting thing, I mean, if you want to understand the impact of any stress quickly, it is very good to observe first chlorophyll fluorescence. Of course, by visual, by our eyes, we can also understand that this plant suffering with the stress. Stress can be an heavy metal stress, I mean more toxic elements in soil. It can be a salinity, it can be a drought. So, but we develop we develop in our lab. This was, we are taking just 5-minute video based on the fluorescence can give very quick results about plant physiology, impact, either it suffering with the stress. We also go for the scan and transmission microscopy to understand the physiology, especially like stomata, how it affecting stomata, because directly connected with photosynthesis. So we wanted to go in deep. If the toxic, the nanomaterial increased in soil, then what will be on plant physiology? And not only those parameters, we also go for the several biochemical enzymes. Undoubtedly, this is not a new trend. When you increase the nanomaterial concentration in soil, it's not new trend that the biochemical have increase or decrease. It's like a similar, like salinity stress, drought stress, even submergence condition. When plants suffer with condition, with any stress, it try to defense, yeah, try to overcome, cope up those stresses. So here we also have the similar situation, and this work we publish in several reputed international journal, because it's, I told you, it's a series of work showing you overview. Because if you increase the, if your soil have more concentration of nanomaterial, if, why are you using if? Because many countries still not using nano enhanced product which contain nanomaterial, especially in agriculture, even here in Russia. But some countries, they are using nanomaterial enhanced product like nano urea, many products. So if this nanomaterial is not biogenic, not biodegradable, because elemental, so some elemental material that might not degrade. So when you are using in first time in your agriculture, like recommended concentration in our study also, we found like 100 ppm, 200 ppm can give good results in wheat crop. So for example, manufacturer suggested some particular amount to your crop, and you using first year, even in one crop you are using 2 times, 3 times, maybe more you are using than the recommended. So the remaining material can be inside your soil, and because of tiny particle, because the companies are, or the producer, manufacturer is not explaining everything, because still we do not have very clear results. If this tiny particle will be in soil, for example, 20 year, 10 year, what will be? Because of this small particle, it, and soil is complex system. There are many other elements, many organisms, microorganisms, so it can interact. So I mean, the repeated application in your field can also remain, exceed the concentration in your soil. So in that case, what will be? So a lot of damages we found in vascular bundles. We found there are, for example, this is normal condition. Normal condition, we have more number of vascular bundle. Vascular bundle, they transferring the minerals, waters. So here, the soil and abnormal. And similarly happen when you go for the subcellular organelles. Subcellular means when you are going like checking the mitochondria size, functionality, nucleus, vacuoles, yeah, plastoglobules, a lot of. So starch granules, so under stress conditions is damaged. All the elements here we see. And we see the subcellular space, there are nanomaterial that accumulated. So abnormality you can see here. Why very big size of starch granules? Because under stress condition, plant was not able to utilize. So, and also the plastoglobulins number decreased, but size increased. Why it is important to study? Because we wanted to understand the mechanism, how internal organelles, cellular, subcellular organelles we have, if the toxicity. Can we avoid this toxicity? For example, we use nanomaterial and shouldn't accumulate in particular parts, shouldn't express, I'm sorry, shouldn't express in essential where we shouldn't express on particular place where we need, where it damages. So for example, we wanted to just enhance the photosynthetic functionality or photosynthetic activity. So our material must express only that place to enhance, to not damage other organelles. And we used recently some biochar and different combination. You can see here, like we use soil. This is the base for our soil bioengineering. So this is 3-year experiment we conducted. First we did in small plastic vessels, then we go for the further, and then we go in a even field condition. So why? Because we wanted to understand if we biochar and bacteria together and treat nanomaterial as a pollutant, then what will we? So if you see in further here, of course, I mean, I am not going in a methodological part. In methodology, we see like soil is spiking is one term. By that you put some elements together, leave it for 1 month, and then you use, so layer by layer we use several these materials, biochar, bacteria, and this bacteria is specific. It's a tolerant microorganism. We isolated because we are in south part of Russia and we have some polluted area, highly polluted. So from that we collected various samples and we go for the screening, and then we found some microorganism. In this case, you see here, this is a clean variant. I mean the soil which is not polluted, but the growth is similar. In this variant, we use microorganism and bacteria, biochar. So biochar alleviated the nanomaterials toxicity because we use nanomaterial as a pollutant in soil system. So all the parameters it enhanced, or it similar with the control condition. So the target was like, if some soil is polluted naturally, like some power plants or some area which is highly polluted, the first target or first challenge to grow any kind of crops, because crop can accumulate and reduce the toxicity, and microorganism can decompose or stabilize, even the convert toxicity to non-toxic, like that. So this was the purpose here also. If we are success to grow in polluted condition, we also not only use nanomaterial, we use bulk material also same time, several sizes of the elements. So here, for example, we are success that growth is quite good. So this approach can we apply as a remediation where have heavy metal pollution. But it's still, again, I mean, it is specific. You cannot use randomly with any kind of pollution. So this work also published like a practical evaluation of integrated role of biochar and nanomaterial in soil remediation process. So here we indicated. Another interesting thing in this experiment that the microorganism functionality increased rapidly where we use bacteria and biochar. Like even I can say like 85% in second year experiment. And this work is also published in environmental pollution. Like we use biochar, metal tolerant bacteria, alleviating nanomaterial toxicity in plant. So again, I emphasize the target is, if this is this experiment target is to grow in field condition where have pollutants, and here we use barley also a bio indicator, somehow accumulator also, but not hyper accumulator. So we can use like mustard. This is methodology. So and even when we going for the all other indexes, plant growth indexes in this condition where we use bacteria biochar, we couldn't see such damages which I saw you previously. You can see here, even where we use bacteria biochar is quite similar. Undoubtedly, sometime you observe in second year like more starch granules. It's also depend, but not such type of damages which I previously showed, where only have nanoparticle toxicity. So this combination of microorganism and biochar, and also not one type of biochar. Here I am just displaying you some experiment. So we use several sizes of biochar. This is also matter because in the beginning we just use coarse biochar, uh, normal biochar, not granules, and we understand that it have good impact. Then we go for the more standardization. We try to use different biochar to see the impact, but when size is more smaller, it have more better impact for microbial functionality, which help for soil natural restoration. And we also go for the detailed analysis, like we going for the each subcellular organelles, to how those subcellular organelles behaving under what damages on that. So this is just cytomorphometric analysis, not needed to read all, but this is important for when you are preparing something to define or to making some product which shouldn't damage all these organelles. This I already showed because this is my recent project. We completed 6 months for this project. We have significant results. We targeting to improve these cycles, you can see here, and we have metagenomic analysis of microorganism of whole Rostov region, and we are trying to use CRISPR. I'm not expert on this aspect, but we have collaboration and in Moscow. So those scientists working how we can modify genome of these rhizosporic microorganisms, and we using like nanomaterial for enhancing the functionality. And biochar I already told because it's a porous material, so it have a lot of benefits to uptake, to absorb, to provide a habitat to microorganism. So this all we trying to work in this work. So the target is to enhance soil for restoration. Because if you increase the functionality of microorganism or density of microorganism in particular soil, so microbial process helping soil for restoration, for decomposition, to produce more organic matter. But this is deficit in current soil. If I am talking about India, the soils are now have very is suffering with the microorganism. So it is necessary to develop a technology by that soil can restore their fertility, productivity themselves to enhance the, the like a nature-like technology. So this is the target for this project. Now, in conclusion, I cannot say you that all these materials are ready to go to the field and farmers can utilize, because still a lot of queries, a lot of relevant information needed to explore scientifically. Like I told you, we do not have still very good approach or low cost technology by that we can produce huge amount of biochar, because this machinery is very specific, because you need to maintain 600 700 temperature. So you need such type of bowler or to create a pressure. So this is expensive, and in lab condition we can produce kilogram 10 kilogram and do experiment. This is different story. But for the farmer or field level, we need huge amount, and for that still we need a low cost technology. Similarly with the nanomaterial also, chemically if you produce nanomaterial, you might be able to produce huge amount. But for biogenic, I mean the using bacteria or using, this is also my work where I suggested the Saunella can be a good candidate for synthesis of biogenic nanomaterial. But in this case, we still need more standardization to produce huge amount to apply in agriculture field. So these are the concern. And also biochar not have only positive impact, it can also have negative impact. Like when you are applying biochar, for example, I discuss and I take biochar here as a soil remediation or soil health improvement amendment. Yeah. But for example, you wanted to use as a crop plant growth promoter, so you are applying in soil, your soil already rich in some nutrients maybe, and then you applied biochar. There are chances the biochar can take or can absorb your essential elements for long term, so that can be a problem. So this all needed to explore, all needed to still investigate what doses, which type of biochar, which type of soils, which type of crops, uh, uh, which type of elements in biochar, how long. Aging of biochar is also another concern. Biochar giving results after 1 year of application. So this all needed to explore before application. Of course, this combination together with microorganism is very effective, but still the microorganism also is specific. Like our condition here in South Russia, in winter like -20, but in summer the temperature is like 40 or even more. So the microbiota is different. You cannot use this approach in everywhere, and maybe not that success. So it needed to explore also the specific microorganism for a specific ecological reason that is necessary to understand. And also some microorganism specific for example zinc, some of them some of them you can stabilize multi-pollutants. So this also needed to combine or needed to think before applying in field condition. So a lot of books I also published on nanobioremediation, biochar, nanomaterial, nanobiochar, nano solution for agriculture, nanobionics. So all these books, edited books, some of them monographs. So here we also try to discuss how effective this technology, more insight, because in presentation it is not possible to say all things. Like if we are talking rhizosphere, there are a lot of information. Hormones, so root exudes, how it playing important role in rhizospheric microorganism, interaction between plant and organism, how we can enhance this interaction for positive way. This all we are working. And also biochar and nanomaterial, which type of biochar, what doses, how long. So all this procedure we are trying to work. So thank you very much. And also I want to emphasize that little bit about our university. I am working since 2000 early at Southern Federal University. This is a central university located 1000 kilometer from Moscow, south part of Russia. So this more than 5,000 scientists working here, and we have only one department. It's a soil and land reclamation department. We have biotechnology, we have several other ecological, and also several other like engineering, economics, business, and so many other. We also have possibility to invite young researchers. Just a few weeks ago, one PhD student from lovely professional university, he completed 3-month training in my lab and he returned back. So there are possibility for the students, they can come and work minimum 3 months, even maximum 1 year they can do research with us in our lab. We also have the possibility for PhD. Now I have 2 PhD students, both are from India. And also we have postdoc possibility, those who completed PhD and wanted to work as a postdoctoral. I also have one, I confirm, but second we are still thinking who we can take, because few weeks ago I announced these positions. So also for the scientific collaboration, we are very open. We can collaborate on this aspect. I am not only one, those who working in this group, we are so many people. Also some of them working on pesticides, some of them working on dye, some of them working on heavy metals, some of them working on different aspect, even GIS and remote sensing, AI and machine learning for agricultural improvement in sustainable way. So thank you very much.
चैट बॉक्स ऑन कर दीजिए रोहित जी। यदि किसी की इस टॉपिक से लेके कोई क्वेरी है, कोई सर से बात करना चाहता है ना, कैरियर गाइडेंस के लिए, तो 10 मिनट हम डाउट क्लियरिंग सेशन का रखते हैं। तो आप चैट बॉक्स ऑन कर दीजिए, या 1 to 1 कन्वे भी करना चाहे, बात करना चाहे सर से, तो कर सकता है पार्टिसिपेंट। ये कैसा इंटरफेस आ रहा है आज, इसको देखो जरा। कुछ लोगों की रिक्वेस्ट है कि सर पीपीटी बहुत बढ़िया थी, पीपीटी चाहिए होगी पढ़ने के लिए।
Okay, will share. I
जी
will
जी।
share, but this lecture maybe you can also find. Thank you very much.
सर आप काइंडली अपना चैट बॉक्स ऑन करेंगे प्लीज, कुछ क्वेरीज उसमें आई हैं सर।
हाँ मैं देख रहा हूँ।
जी प्लीज।
जी। बाकी कोई भी अगर आना चाहता है ट्रेनिंग के लिए, चाहे बैचलर स्टूडेंट हो, मास्टर स्टूडेंट हो, पीएचडी स्कॉलर हो, जैसे पीएचडी स्कॉलर की कई बार डिमांड रहती है कि कैन वी डू ट्रेनिंग फॉर 6 मंथ, और कैन वी डू आवर ओन एक्सपेरिमेंट, इस तरीके का, तो देन वो आ सकते हैं। ऑफ कोर्स वी ट्राई टू सपोर्ट देम, वी हैव फ्री लिविंग अपार्टमेंट, एंड व्हाट they need to support airfare by themselves. जैसे इन्होंने लिखा है कि सैंडी सॉइल में बायोचार डाल सकते हैं क्या, ये प्रॉब्लमेटिक होगा? नहीं, बिल्कुल भी प्रॉब्लमेटिक नहीं होगा। इवन बहुत सारे एक्सपेरिमेंट ही सैंडी सॉइल में ही करते हैं, क्योंकि बहुत सारे अदर वो एलिमेंट्स नहीं होते। तो सैंडी सॉइल में नहीं है, आप नैनो बायोचार डाल सकते हैं। अब बायोचार किस का सोर्स क्या है, व्हाट इज द सोर्स मटेरियल फॉर पायरोलाइसिस ऑफ दैट बायोचार, ये बहुत डिपेंड करेगा। बाकी जैसे अभी जो रिसेंटली वी आर वर्किंग लाइक मोडिफाइड बायोचार, तो मोडिफाइड बायोचार में वी आर यूजिंग लाइक डिफरेंट डिफरेंट एलिमेंट्स, उनके साथ मॉडिफाई करके फिर हम सॉइल में अप्लाई कर रहे हैं। देन वी आर आल्सो ट्राइंग टू सी अगर बायोचार का साइज छोटा बड़ा करके देखते हैं तो क्या इम्पैक्ट पड़ता है। तो वी हैव रिजल्ट्स ऑन दिस।
वैसे सर लेक्चर तो अपने आप में बहुत सेल्फ एक्सप्लेनेटरी था। हमें नहीं लगता कि बहुत ज्यादा क्वेरी की आवश्यकता है पार्टिसिपेंट्स को। कुछ लोगों ने बस वही अपनी इच्छा जाहिर की है कि आपके साथ काम करना, आपकी लैब में ट्रेनिंग करना, पीएचडी पोस्ट डॉक करना। तो सर हमेशा लिंक्डइन पे अवेलेबल रहते हैं। सर की एक ईमेल भी है, आप लिंक्डइन पे भी सर से बात कर सकते हैं। बहुत एक्टिव रहते हैं, बहुत एक्टिव पर्सन। मैंने कभी किसी हेल्प के लिए सर से बोला होगा तो हमेशा सर ऑलवेज भी देयर सर। तो मैं
थैंक
अपना
यू।
व्यक्तिगत अनुभव में, पर्सनल अनुभव बताना चाहूंगा कि हमेशा हमारे लिए सपोर्टिव रहे। तो कोई भी स्टूडेंट, कोई भी फैकल्टी, कोई साइंटिस्ट लिंक्डइन के थ्रू बात करना चाहे, मैं ईमेल भी प्रोवाइड कर दूंगा, आप ईमेल के थ्रू बात कर सकते हैं। थोड़ा बिजी शेड्यूल रहता है तो कृपया ऐसा करें कि व्हाट्सएप पे अप्रोच ना करें। लिंक्डइन पे भी आप रिप्लाई करते हैं सर, और ईमेल पे भी बहुत जल्दी रिप्लाई करते हैं। तो बहुत बहुत धन्यवाद सर। आज आपकी एक
थैंक यू सो
मीटिंग भी थी,
मच।
बहुत इंपॉर्टेंट मीटिंग थी,
थैंक
है
यू
ना,
सो मच।
उसको आपने
अभी
रिशेड्यूल
है
किया, है ना, मेरी
आज पूरे
रिक्वेस्ट पे
दिन।
आपने रिक्वेस्ट को एक्नॉलेज किया,
थैंक यू
है
सो
ना।
मच। थैंक यू डॉक्टर अंकुर,
आप
रियली ग्रेट प्लेटफॉर्म फॉर स्टूडेंट्स, फॉर अस आल्सो। इट्स ग्रेट प्लेजर टू कनेक्ट एवरीबॉडी।
हेलो विष्णु सर।
थैंक
हेलो।
यू रोहित। हाँ प्लीज।
सर, like I have a question regarding biochar.
हाँ
हाँ हाँ
सर
रोहित
एक्चुअली
प्लीज।
वर्किंग सर मेरा पीएचडी उसी पे कर रहा हूँ अभी। तो सर मैंने कोटेड मटेरियल किया है बायोचार का, लाइक यूरिया पे कोटिंग किया है, उसको एनरिच भी किया है, देन एक्टिवेट करके भी कोटिंग किया है। सो
हम्म।
उसका सर उसी पे अभी कुछ देखे रिजल्ट्स आएंगे नहीं। तो सर आपका कुछ इस पे आइडिया, कुछ मतलब किस टाइप का और इम्प्रूवमेंट कर सकते हैं इसमें?
अगर आप कॉम्बिनेशन लेते हैं, मैं फिर सजेस्ट करूंगा, जैसे
जी
नैनो
सर।
कोटेड बायोचार, ये ट्रेंड में है, हमने भी किया है।
जी
इवन
सर।
गौरव के साथ हमारा डिस्कशन हुआ था फ्रॉम
हाँ
एलपीयू।
सर, गौरव से भी मेरा बात हुआ था तो चंद्रा सर भी
हाँ
बोले तो इसी चीज पे
हाँ,
भी
ही
काम कर रहे
वांटेड
हैं।
टू वर्क। मगर अगर आप एक कॉम्बिनेशन माइक्रो ऑर्गेनिज्म के साथ लेते हैं, कोई माइक्रोबायोलॉजिस्ट हो अगर आपके डिपार्टमेंट में, देन इट
ओके।
विल बी रियली इंटरेस्टिंग कि एक 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 पैरामीटर आप लगा के देखिए माइक्रो ऑर्गेनिज्म के साथ, माइक्रो
ओके सर।
ऑर्गेनिज्म प्लस नैनो कोटेड
बायो
यूरिया बायोचार,
कोटेड बायोचार।
कोटेड
ओके।
बायोचार। तो कोटेड का
ओके
अभी
सर।
काफी मतलब ट्रेंड में है। अभी लॉन्ग टर्म इसका क्या इम्पैक्ट होगा, कैसा होगा, इसका आइडिया नहीं है, क्योंकि ये रिजल्ट्स अभी नहीं है। पर
जी सर।
इंटरेस्टिंग है इम्पैक्ट। हमने भी किया है, हमारे जैसे रिमेडिएशन का ज्यादा हमारा वर्क है। पर एग्रीकल्चर में भी, जैसे सस्टेनेबल एग्रीकल्चर के लिए जो फार्मर है, तो वैसे तो डिफरेंट डिफरेंट कंडीशन है। रशियन जो फार्मर्स हैं, कि हर एक फार्मर लगभग 3,000 4,000 हेक्टेयर लैंड उसके पास होती है, तो दे आर
हम्म।
फुल्ली मैकेनाइज्ड, उस तरीके से एग्रीकल्चर वो करते हैं। तो उनके साथ हमने कुछ एक्सपेरिमेंट, आई मीन हमने मटेरियल दिया कि प्लीज आप टेस्ट करिए अपने फील्ड में, तो वो लोग
यस
उस
सर।
पे वर्क कर रहे हैं,
वो
पर
तो सर
इंटरेस्टिंग
हमने भी लाइक इट्स लाइक अभी हफ्ता हुआ तो फार्मर्स को लाइक 3-4 टन के आसपास दिया वो कोलेबोरेशन मतलब बायोचार उनको बना के। सो लेट्स सी, लाइक फर्स्ट टाइम यूज कर रहे हैं, अच्छा रहेगा तो आगे और चीजें जाएंगी।
नहीं, मैंने देखा गौरव ने जो बताया वो टेक्नोलॉजी, तो
यस
हम
सर।
भी थोड़ा उसको एक्सप्लोर कर रहे हैं, क्योंकि अभी तक तो हमने जो पायरोलाइसिस कंडीशन है तो उसको 6 और 700 के बीच में ही किया है। और उसका
जी सर।
उस सिचुएशन में मुझे नहीं मालूम कि टेम्परेचर कितना है, जैसे आप वो जो लोकल उसका वो कर रहे
सर
हैं जैसे।
400 उसमें हमने मेजर किया था, लेकिन 400 लाइक 400 अप टू 400 लाइक
हम्म
मेंटेन रह रहा है वहां पे वो, अप टू
हम्म
400 लाइक
क्योंकि
वही
कई बार ये
उसमें
भी
है।
होता है ना कि हम अगर बर्न कर दें और वो बायोचार बनेगा क्या, वो सब डिफरेंट
ऐश
अप्रोच।
में।
हाँ
यस सर, यस सर।
ठीक है, तो थैंक यू सो मच। इधर
थैंक यू
भी
सर, थैंक यू सो मच
हाँ,
फॉर योर
ठीक
गाइडेंस, थैंक यू।
बहुत
बात
जल्द
है।
हमारे पार्टिसिपेंट्स को इंटरनेशनल कॉन्फ्रेंस में भी सर का एक लेक्चर मिलेगा फरवरी में, मैं
थैंक यू,
आशा करता हूँ। जी जी, बहुत बहुत
थैंक यू
धन्यवाद सर।
अंकुर, थैंक यू
जी
सो
जी।
मच।